Okay, this is part 2 of the Microscope Lab. Is that if we were doing this in the lab, this would be to get you used to using the microscope with the 3 different powers. It's also a bit of a preview for something we're going to use during the diffusion lab later in the semester. So I'm going to set the slide up here. I've got a slide that I've cleaned and dried off, sitting on the desk. I'm going to take a drop of water out of my little big beaker here, put a drop there. I've got a plant in here that we're gonna make sing and dance when we do the diffusion lab, but right now we're just gonna look at a leaf. So I'm gonna grab the stem, if I can figure out where the stem is, and pull off one leaf. Put the leaf in the drop of water. I have a cover slip that I've also cleaned and dried. Gonna put that over the drop of water. And then we're gonna go into the microscope. I'm gonna turn the lights down while my wonderful cinematographer sets herself up for this. Remember, this field is much brighter and whiter under the microscope than what you're seeing up there. That's as bright as I can get it, and it still looks yellow. But it should never look yellow when you're using the scope. Okay, so. Where's my focal labs? Okay, why is this not picking up anything? I guess I should have checked this. Nothing. Let me see what I've got here. Ah, slightly off. Nope, that's. Ah. Is nothing? Okay, I do not understand. I may need to change the mode here. There we go. I don't work with this very much. Always fun. Okay, I just turned down the light a little bit. And I'm going to change the iris diaphragm to mess with the contrast to get the edges to show up. So in part 2, you're going to do me a few drawings. This is normally to prove to me that you can focus it at different powers, but here you have to depend on me to be able to focus it at different powers. This is what the leaf looks like at, that's a bad place to put the light. The light control is a dial, and it's right above the fine focus knob. It's amazing how badly they design modern microscopes. This is what the leaf looks like at low power. The round thing is a trapped air bubble. I'm just adjusting my microscope so that it's pointing at both of my eyes at the same time. I think it is. And for this part of the lab, you just draw me a little piece of the leaf. Just draw me what it looks like at this power. Just to give you an idea, the little brown spots are fungal spores. They're on the leaf, but they're not in the leaf. And the leaf does have a little ridge up the middle, but it's very much an algae, so it's a lot of simple, similar cells. Now the next thing we're doing is moving up to middle power. This is a good scope. It's pretty close to being in focus at middle power. This is what it looks like. I'm gonna move up. Oh, there goes something swimming by. A little protozoan on the surface of the leaf. Yeah, this is not binocularing right. Yeah, I should have picked a leaf that wasn't quite this green. These are so packed full of chloroplasts that it kind of makes it a little muddy. This is a better area. So here, you're gonna draw a little bit. Notice as I change the focus that the thick edged rectangles look like they have very light lines through the middle of them. That's gonna become important when There's the little lines. That's gonna become important when I go to high power, which is what I'm about to do. So, like a good microscopist, I lean back and I watch the lens go into place, make sure I don't hit anything. If it's a good scope, it'll be close to being in focus, and it is. There's one of the little creatures going by.
You can hardly see it on the screen.
Yeah, well, I can hardly see it on here. It's very transparent. Being transparent is very good when you're a little creature and things kind of come up behind you and try to eat you. Now, the question you would get is not to draw the picture. Okay.
Yeah, that's what I'm saying, that you could hardly see it. You could hardly see the leaf.
Okay, I don't quite understand why you're not seeing anything.
And if you see a little bit,
Okay,
It's just-
something's very
it's
wrong.
like light. I think it's a light issue.
Ah,
there.
okay.
Yeah.
Thank you. What I see and what you see, I'm trying to match up. Now this is in focus at high power. All those green dots are chloroplasts. They're the photosynthetic organelles inside the cells. Now, one of the things you'd be asked to do is, using the focus, try to figure out how many layers of cells there are in this leaf, which means, okay, I'm gonna change the focus. See the edges go out of focus? See the new edges come into focus? There's edges here. The second layer, the cells are much thinner edged than the ones that are now just shadows. And if I keep changing the focus, it's just gone. It's not in the leaf anymore. I'll come back through. We hit these fine edges. We hit these thick edges, and I'm gonna keep focusing in the same direction. This is just going through the chloroplast, they're fat cells, and it's gone. So I'm gonna ask on you to count, how many layers was that? How many layers were in focus as I focused through the leaf? And that is the last question for this part. As you can see, this is really a how to use the microscope kind of part. But I wanted to preview these cells because we are gonna look at them during the diffusion lab. And that's it for part 2. Oh, you want me to focus it better? Oops. Focus.